This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1107, recorded on April 16th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. And it is a gorgeous, gorgeous spring day here. 67 Fahrenheit, 19 C, few puffy clouds in the sky. It's supposed to be nice tomorrow, and then a little more rain rolls in.
Here in Chelsea, it is 21 Celsius, which must be like 69-ish, right? Or 70-ish. And sunny. Also joining us from Madison, New Jersey, Brianne Barker.
I have 70 Fahrenheit and a beautiful sun. It was gorgeous yesterday. I think we get the rain starting tomorrow. But it's lovely here, and it's great to be here with you all. It's also very cold in my office.
Oh.
It would be much nicer to be in that sun.
Is the AC on?
The AC is always on in this office.
I went to teach yesterday in campus. Everybody's outside. And hardly anyone came to class. There were like 20 people in class. There were 100 students in the class. 20 people showed up.
My students were very confused why I didn't go sit on the lawn to do my paperwork yesterday. And I was like, sorry, guys.
I can't teach outside. I have a lot of slides. Well, if you like what we do on these programs, these science programs, we'd love to have your support so we can do more. You can go to microbe.tv/contribute, and your contribution, at least in the U.S., is federal U.S. tax deductible because you make the contribution to Microbe TV, which is a 501c3 nonprofit entity. And so if you want to hear good science, microbe.tv/contribute. By the way, this is Tuesday that we're recording. It's an unusual day, and thank you to Brianne and Alan for agreeing to do that. The reason is I'll be in Princeton on Friday working on the textbook, Thursday and Friday. And so that's why we're Tuesday. But you don't know it except that I told you. You wouldn't know because it's going to sound the same. We have a public service announcement for ASV, American Society for Virology, the annual meeting. The early bird registration ends on April 22nd. That's Monday after this drops.
Yeah, so if you're listening to this Sunday night right after the podcast drops, you better get your registration in immediately. And if you're listening to it later in the week, you're too late. You're going to have to do regular registration.
Yeah, you pay more.
Yeah.
And also consider registering for satellites. You can add them even after
I've always wanted a satellite.
Already registered. What kind would you like? You want a Sputnik, the cute little silvery kind?
I'd like something that's got a radio transceiver on it, maybe a camera, some sensors.
Then the modern ones they're all just solar panels, big things, right? But if you want your own satellite, it's a meeting at ASV.
Ah, right.
You can add a satellite to your registration record. Just use the email you registered with and the reference number that you will see in your initial confirmation email to log in. And if you register for one satellite, you can go to any of them. You could move around. It doesn't really matter. And here are the satellites for this year. So it's a day before ASV begins.
Vincent, after you read them, you should guess which one I'm going to.
Okay, I will. We have invertebrate virology, unique and conserved aspects of virus-host interactions. A veterinary viropalooza, improving the control of viral diseases of animals. Studying bats using a One Health lens, bridging the gap between bat virology and disease ecology, virology in a global society, and navigating virology funding opportunities with NIH, NSF, and USD. So I would guess the bat one.
I'm going to the bat one.
Yeah.
I was going to guess bats or virus-host interactions.
Yeah.
But bats would be, that's going to be cool.
Yeah, I might go to that one. I registered for a satellite. And I probably go to the bat one also, yeah. And don't forget the International Conference, Pandemic Preparedness, Achievements, Current Challenges, and New Frontiers, November 11th through 13th in Trieste, 2024. I would love to go to that. Just because Kathy said Trieste is a good meeting venue. And the purpose of this conference is to give an overview of known and emerging viruses and their potential epidemic risk, to foster knowledge sharing and networking, encourage the development of broad synergies and multidisciplinary cooperation. It'll be about diagnostics, epidemiology, surveillance, drug discovery, and preventive approaches with the aim of implementing synergistic, innovative, and effective response strategies to tackle the challenges of re-emerging infectious diseases and respond to future pandemics. And there's a website that we will put a link to in the show notes so that you can go register. Let's have a look. International Conference, Pandemic Preparedness.
The pictures look pretty.
Yeah, except all the cookies in Italian, because Italian cookies are good. Trieste looks very nice. Look at those cool buildings. It's 208 days from now. And they have pictures of all the speakers. Very cool. All right. Check it out. I don't know what I'm doing in November. I'll have to look. What else do we have here? Don't forget Dixon's book, The New City, at despommier.com. And he's also has a blog post about it, which we'll link to in the show notes. So I just want to tell you, now that you've been sucked into this episode, that it's going to be all email. Click,
Yep.
click, 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 click. Now, don't leave.
There they go.
It's science. You're going to learn science. They're good questions.
It's going to be your questions.
Your questions. And we've had a backlog for a while. Sometimes we don't get to them.
You never know what kind of banter will come up.
Well,
Yes.
that's the thing. The banter surrounding the questions is fun. We used to do these all the time, in fact. And then we did a couple during the pandemic years and then stopped. And now, because it was quick turnaround between Friday and today, I thought this would be the easiest on everyone instead of having to find a couple of papers and read them. So here we go. There are a lot here. These go back to January, but they're fun. So let's start with Brianne.
All right. Anish writes, Dear TWiV team, I hope this finds you well. I'm Anish, and I'm currently enrolled in BI 115 at Caltech. As part of our course, we've been encouraged to engage with external resources to deepen our understanding of virology, one such example being the TWiV episodes. I'm writing to submit a few questions and insights for your consideration, which were inspired by the most recent episode. I've also CC'd Camille to highlight this initiative for extra credit for our course. So question 1. In the most recent episode, the strategy of stabilizing viral spike proteins with proline substitutions was highlighted, particularly in vaccine development. Considering the diversity of spike proteins across different viruses, how do you see this strategy evolving to tackle emerging pathogens? Are there specific viral families or pathogens where this approach might be particularly impactful but hasn't been explored yet? Should I keep going? Should I do all of them, or do you want to answer in between?
Let's do them one at a time.
Okay.
Yeah. This must refer to the Jason McClellan episode.
Yes, that was my assumption.
You know, we can be safe and say it'll work for some and not others.
Yes.
Right.
I mean, the key is that so the prolines prevent the conformational change. So the key is where that usually happens, right?
Right.
So for spike of SARS-CoV-2, it happens after a proteolytic cleavage event at the cell surface.
Right.
And so if you make spike protein as part of a vaccine, that would happen there. But if it happens only at low pH in an endosome, then I don't think it would matter, right?
Yeah, I was wondering when reading the question about whether this would be particularly useful for those that involve proteolytic cleavage and perhaps not for those that had conformational changes for other types of things. I think the only other thing I could imagine is how much some of this might change particular epitopes. If there is an epitope that is particularly important either in the stabilized or unstabilized form, that would impact this as well. And so I cannot name every epitope of every virus spike protein, nor can anyone. So I don't know that we can give examples, but I agree with it's probably useful sometimes and not useful sometimes.
Yeah.
So I think for flu, it wouldn't be useful because it's low pH conformational change, right?
Mm-hmm.
So the ones that happen at the cell surface, I think they would be the ones, and that would be coronaviruses and paramyxoviruses, to name just two.
And this is something you'd only do for enveloped viruses, because if you don't have an envelope, then you need to generate antibodies against the capsid, which is a different problem.
And in fact, that jives very well with this episode, which is all about envelopes. Right?
Yes,
Exactly.
May I
opening
have the
envelopes.
envelope, please? Didn't you have a title once of something with envelopes?
Probably
think I may
something
have
like
used that
pushing
in
the
it.
envelope.
Yeah, we may have used that in an email episode.
Question 2. The conversation around leveraging the S2 subunit of the coronavirus spike protein to potentially craft broad-spectrum vaccines caught my attention. Given the noted challenges in generating potent neutralizing antibodies targeting S2, what are your thoughts on enhancing their efficacy through combined strategies like nanoparticle display or novel adjuvants? Could such a multifaceted approach lead us closer to pan-coronavirus vaccines? I would say, sure, definitely. I think, I can't remember the exact details of this course that you are writing us about. We've had people write from this course in the past, but I remember correctly, perhaps Pam Bjorkman might be involved in this course, and she talked to us on TWiV about an approach just like that that seemed really promising. So your professor might, or someone your professor knows, might be involved in exactly that.
Yeah, making nanoparticles with multiple spikes, right? I remember that approach, yeah. And also Jason talked about different approaches like introducing disulfide bonds on subunits and dealing with pockets. Yeah. So I was really impressed by the creativity, different approaches. Don't just make a protein and stick it in a mouse and see if it works, but you try and engineer it in a way based on the structure, which makes a lot of sense. We might as well take advantage of that information.
Yeah, and you can certainly.
Go ahead.
I was going to say you can certainly also think about the structure of the antibody and the antibody binding site and try to think about how to make sure that your antigen is complementary.
Yeah. And the trick here, of course, is to generate antibodies against something that people don't normally generate antibodies against, so we don't really know what is going to be the best way to trigger that.
Absolutely. Question 3. How do you anticipate vaccine design strategies will adapt to keep pace with rapid viral evolution? Are there general lessons from the experience with SARS-CoV-2 that could be applied more broadly to anticipate and counteract variants of other viruses? Or is the evolutionary dynamic of SARS-CoV-2 unique in its challenges? Anish also says, thank you for considering my questions and your continued effort to demystify the complex world of viruses for listeners worldwide. Well, Anish, on Friday, we talked about how great T cells are at recognizing heterologous versions of the virus. So my first thought as I see this is vaccine design strategies should include some mechanisms to induce T cell responses and not just antibody responses, as that may give you broad protection. I also have been constantly impressed by some of the work that I've seen during the pandemic where people have really looked at what goes on in the germinal center as antibodies may broaden to be cross-reactive as responses go on. And I think the more we learn about that, the more we might be able to take advantage of it. But I certainly learned a lot during this time about that. So I'm guessing other people might have too.
I think that we have, the immunology, we've learned a lot, but I also think that, the mRNA vaccine is the first generation. I think there's going to be improvements. I was reading recently about these self-replicating RNA vaccines where you take an mRNA, same idea, but you put in a polymerase of a virus, so it's produced and it replicates. So you don't have to give such an initial dose, right? You give a lower dose and you have fewer side effects, and then once the RNA is in the cell, it replicates and makes more protein. I think that can be very interesting. To modify.
Yeah, and given the side effects a lot of people have experienced with the mRNA vaccines, I think anything to address that would be very helpful.
Maybe changes to the adjuvants.
Yeah.
Okay, Alan, can you take the next one?
Sure. Adam writes, in the latest Offit episode, Dixon described some interesting astronomical phenomena, after which you had extended discussion. A couple of corrections. The black hole at the center of our galaxy is 4 million times bigger than the Sun, not billion. The sun will end its life as a white dwarf, not a neutron star or black hole, no supernova. Third, we actually have a couple of people who wrote in about this. The solar system orbits the galaxy at 220 kilometers per second, so kind of fast. We're not likely to hit anything. Next, gravity doesn't suck. It's an attractive force, but it doesn't pull things in. Planets orbit stars for a long time, staying at the same distance, and stars orbit black holes the same way. Finally, if stuff falls into the black hole, it never comes out. However, if the stuff falls very close to the black hole, it can be launched out as a jet. Dixon got a lot more right than he got wrong, though. I'm just nitpicking. And Adam is an associate professor in the Department of Astronomy at University of Florida, Gainesville. Thank you.
So what does this mean? Just because we're going fast, we're not likely to hit anything? I don't understand that.
I think some of
Space
it's
is really, really empty.
And other things are moving too.
Yeah.
220 kilometers a second, like 1001. It's 220 kilometers. Oh my gosh, it's like upstate New York, right?
Yes.
It's just incredible. Wow, this is cool. I wouldn't have known any of this. Thank you very much, Adam. Bo writes, a few episodes ago, the hosts were talking about mammals in Australia. There's get a theme here that we're being corrected a
Yes.
lot, right?
Yeah, one of the things in our letters often comes up.
Well, when we go off topic, right?
Yeah.
That's the thing. There are native bats and rodents in Australia, according to fossil records from 15 million years ago and 10 to 5 million years ago. They are the only placental mammals native to Australia. By native, I mean they were not brought there by humans. I was astonished to find out about Australian bats myself a few months ago, so I thought it was worth sharing. Thank you for your hard work and, as always, excellent content.
And by fossil record, you mean they're not around anymore, I assume.
I guess. Yeah, they lived millions of years ago. All right.
Very interesting, though.
Yeah.
The placental mammals did make it there.
Brianne.
David writes, supernova with a capital no.
Oh, that's so clever.
Just a small correction for Dixon. Our sun will not go supernova. It lacks the mass and energy. It will likely expand to a red giant, then shrink to a dwarf star. Of course, we won't be around to empirically prove this. However, our knowledge of stellar physics tells us that this is how it will die. I really like that people like to write in to correct us, because how else am I going to learn my physics?
No,
Yeah.
it's totally fine. I have no qualms about it. Even you can correct us on our virology. It's fine. That's the thing scientists should do, is admit when they're wrong, right?
Absolutely.
Alan.
Martin writes, Dear Twivers, I have only just learned from David Attenborough's TV series on mammals about the bats in Austin. Any comments, Rich? Well, Rich is not here, but I can comment on them, and they were a spectacular thing to see when we were down there for TWiV 500.
Oh, you went too?
Yeah, went to the park by the bridge, and
cool.
sun goes down, and all these bats come streaming out from under the bridge. It's very, very cool.
A lot of people there too.
Yeah,
right?
oh yeah, there's a big crowd.
It was like an event. And the funniest thing was
It's like Sunset in Key West.
They fly out, and then this guy in front of me, he looks up and he goes, "Is that it?"
Yeah. Some people just,
It was like 500,000 bats that just flew out. I mean,
oh
come
yeah,
on,
yeah. You see this every day. Okay, fine.
And Randall writes, a couple of months ago, I sent you an article about the new virus, Alaska pox. I remember wondering at the time if any other states had viruses named after them. Well, Alaska pox is now boreal pox. In keeping with the trend to not name viruses after geographic locations or animals, the name has been changed. Okay, so here's a link to the article on the Alaska Beacon. Alaska pox no more. Newly discovered disease and virus is to be called boreal pox. Is it boreal or boreal?
Boreal,
pox?
I think.
Boreal. The disease and virus exist in the environment well beyond the state's borders, making the new name more scientifically accurate, officials say.
Mainly
Okay.
the issue was that initially they thought it was just, a rodent virus, and then it turned out it could infect people, and they said, yeah, we don't want it named after our state if somebody can get sick from it.
Yeah, I always find this strange, because we're not going back there. How do the people near the Ebola River feel, or near the Haunted River feel?
Why don't you? Brianne, take Braden.
Braden writes, Dear TWiV team, thank you for your podcast. I've been a regular listener since 2020 and came dangerously close to applying to an immunology master's program as a result of wanting to introduce people to the truth about vaccines by osmosis with a heavy textbook applied to the head. I get it. I felt like my questions, whether autism cure, in quotes, diets actually reduce symptoms, or if avoiding inflammatory foods like wheat freed up people's immune systems and allowed them to mask better as neurotypical, weren't exciting enough to justify Strasbourg giving me a double master's spot. I applied to programs closer to my undergrad degree. My question. In a recent episode, someone, was it Vincent, said that cancers we currently think of as not being caused by viruses are cancers caused by viruses we haven't found yet. My aunt, my dad's sister, recently passed from pancreatic cancer. This is the same cancer that claimed my dad's brother and 40-something years ago, my dad's dad. As far as I know, my grandfather wasn't diabetic, although both my dad and both of his siblings were. Dad is still alive. Dad's mom died of a heart attack in May 2020 after having an antibiotic-resistant respiratory infection with fever and headache that started in November 2019 and progressed to antibiotic-resistant pneumonia. I don't say that she died of COVID near my anti-vax conspiracy theorist relatives, but it seemed like she was an early case of that. Do you think it's possible that my aunt, uncle, and grandfather, and maybe my cousin's brother and I, have some kind of endogenous virus that passes through sperm cells? Or is it just a genetic predisposition to diabetes, pancreatitis, and an inability to eat a lot of fatty, sugary foods? My gran was a legendary cook and made a lot of cakes and pastries. How she died of a COVID-like disease in her 90s and not diabetes remains a mystery. Unfortunately, because she died in hospice of a known disease, nobody biopsied my aunt's pancreas before she was cremated. I'm looking forward to the upcoming sports science podcast. My undergrad was in sports science and ergonomics, and the department, Human Kinetics and Ergonomics, Rhodes University, is still active in sports research. Candace Christie and Jonathan Davey are publishing in the sector, Candace more than Jono. Keep up the great work. You've been my lifeline of sanity for the past 4 years, and I'm always disappointed when people don't take my recommendations. I was the person who emailed President Macron to recommend you and was quite overjoyed that he stopped recommending hydroxychloroquine as his COVID policy. Best regards from warm, sunny Zimbabwe, Braden.
Emailed President Macron. That's pretty funny.
I think that's great.
You should listen to TWiV, President.
Thank you for emailing President Macron. I like that. I think that there are definitely cancers that are caused by viruses we don't know about. I don't know if I would say every single one, or every single cancer is caused by a virus we don't know about. But I think that there definitely is an underrepresentation of how many viruses are causing cancer.
Yes.
Yeah, I mean, I think people nowadays grind up tumors often and they sequence the genomes, right?
Yeah.
So you would be seeing...
There's a whole project of that, the Cancer Genome Atlas.
Yeah. So I think we would see if certain viruses were always associated or largely associated with certain cancers, right?
Assuming they're known viruses.
Yeah, it's true.
Yeah.
There's a lot of dark matter.
Yeah.
Yeah.
I do not know about endogenous viruses and diabetes, though there have been immunological links with some other viruses influencing tolerance and diabetes. I don't know of any of those being endogenous.
Yeah, I think enteroviruses have been implicated along with genetic predisposition.
Yeah. Yeah, and it's not really ever panned out as a thing as opposed to them being in a lot of people. It's hard to know.
Mm-hmm.
Okay, Alan, you're next.
James writes, Hello, in listening to TWiV 1095, Monkeys Fly and Mice Exaggerate, Dixon only got it half right on platypus. Females lose their spurs in adulthood, and males basically use their spurs for fighting other males. Be sure to watch this truly heartwarming tearjerker, and sends a video which I have not watched yet. This is from PBS, The Platypus Guardian. Guy befriends a platypus. Okay, cool.
Heartwarming sounds good, but tearjerker, I don't know.
Yeah.
I don't want to see sad things
Sad
about
platypus
animals.
story.
They have enough problems. Okay. Jay writes, Dear TWiV, TWiV 1105 is an excellent episode. I guess that's the last one we did, right?
Yeah, I think so.
I would like to point out one minor correction. At around 128, Vincent comments that common cold coronaviruses don't make anybody die outright. But common cold viruses can, in fact, be fatal. Mortality may be low, but I've seen it myself in immunocompromised hosts. A quick search of the medical literature provides plenty of other examples. Here's one summary. Quote, the disease is more severe and dissemination is more likely in patients with impaired immunity. For example, organ transplant recipients, HIV infection. Fatality rates for untreated severe adenovirus pneumonia or disseminated disease may exceed 50%. And the link there. Adenoviruses are DNA viruses that typically cause mild infections involving the upper or lower tract, the GI tract, or conjunctiva. Rare manifestations of ad infections include hemorrhagic cystitis, hepatitis, hemorrhagic, ... I love learning with all of you. Keep up the great work. Jay is an MD in Los Angeles. So, yeah, I should never say never.
Well, those are adenoviruses he's citing, so I would point out that it's not common cold coronaviruses.
Yeah, I'm sure they kill some people in there, but the general effect of common cold coronas and rhinoviruses too is not to kill you, right?
Yeah.
But everything is relative. It's just more likely not to kill you, but it's never zero.
And if you're having a lot of health problems and other challenges, and then you get a common cold coronavirus, that could be something that pushes things over the edge. So maybe it's not the thing that killed you, but it could be a thing that killed you.
And yes, so there you go. Correction, right? I like that. Let me take the next one because it's very short and it has to do with me. This is from Tom. Dear Vincent, Richard Ebright makes public the names of people he considers deceivers. This is sad, actually. Someone sent, Tom sent it, not someone. I cut off the rest of it. He said, I hope this doesn't make you a target for attacks. No, nobody cares.
Yeah, so this
matter.
is something this Richard Ebright posted on Zitter. And it's a
Zitter.
thread of
That's a nice way to put it. I like
it.
I haven't heard
mean,
that before. I like that.
I'm getting so sick of the X, the platform formerly known as Twitter. Just call it Zitter.
That's really good.
And I'm actually I'm a little upset about this. I'm not on this list, and I'm
You want to be on it? Is that why? You're upset?
Yeah. If you're gonna name prominent people who you consider to be deceivers, come on.
So what the deceivers, scientists who refuse to acknowledge that SARS-CoV-2 came from a lab, right? That's what his problem is. He says the overwhelming evidence is that it came from a lab.
Which
it's not
is
true.
180 degrees wrong.
No, it's wrong. And so he says people that speak out against this is not the spirit of science,
No.
Dr. Ebright, at all.
No.
And, people disagree all the time. And for you to publish a list of people who disagree and call them deceivers is just, oh my gosh, it's so abnormal.
It's propaganda.
Mm-hmm.
What is he getting out of this?
He's talking to his base. There's a group of people who are just off in this little echo chamber, and that's what they want to hear.
Yeah, I don't understand a lot about that base or why they want to hear. I find a lot of it confusing. I am also not on this list, though I have been on similar lists and gotten the DMs on Twitter, or what was then known as Twitter, because of that. And at one point, some of this came up with some administration people, and they said, we think this sounds like you're doing a good thing.
Yeah.
It sounds like you must be saying the
Yes.
right things if you're making these people angry. So maybe you can think of it as a badge of honor, Vincent, that you are prominent enough to be on the list.
Yes.
Well, we think it's because of TWiV. We often
Of
talk
course.
about it here.
Yeah,
So he
sure.
just lists one name, but, I know there's another reason, but I can't talk about it. But I just very sad. And, there was recently a letter to science by a bunch of these people who complained about his behavior. You probably saw that, right?
Mm-hmm.
And his behavior is really not nice, right? If you want to disagree, just disagree civilly,
Right.
not like this. Thank you, Tom. Tom is in Pasadena. Let's see. So that since I did this one, that means we're back
Brienne's
to
up.
Brienne. Yeah.
Yes. Charles writes, "Hello, Twivers. Wonderful day in central North Carolina. 86 Fahrenheit and sunny. One of those days I would like to take off from work." Also, get that looking outside. If you get a chance to see another total eclipse, suggest you look for a clearing in the woods. Your ears will be almost as delighted as your eyes. The birds and the bugs go through their night-day-night cycle in just a few minutes. The linked pictures are from the 2017 eclipse. They were taken at a small electrical substation in South Carolina. One image is just a zoomed part of the other, and he links 2 photos. Then he also says, "These are just pictures I really like. One of huge driftwood and one of IR just outside of Lexington, Kentucky, as well as one from IR from near Edisto, South Carolina. Thanks, Charles. P.S. If you decide to read this during the podcast, I hope Dixon is there. I enjoy his photography." I agree very much with Charles. I was in a pretty rural part of upstate New York when I saw the total eclipse. And he mentions the birds and the bugs. We noted how, as it was starting to get dark, the mosquitoes came out in full force.
You're kidding.
The way that they often come out at dusk. I have mosquito bites like it was the evening in summer that I hadn't had. And I took a video during the eclipse. I was trying to show the 360-degree sunset effect. And I grew up, this is my parents' backyard. So I guess I didn't really think about sort of the nature in the area, but the frogs were going nuts. And every person I've shown that video to here has found the frog noise in the background to be the most dramatic part, more so than the 360-degree sunset or the total eclipse that you can see. Everyone is like, listen to those frogs.
Yeah.
So yes, I would agree. Going to somewhere out in the middle of the woods or in nature was pretty exciting. I recommend it as well as Charles. And don't get bug bites. Bring some bug spray.
Yeah.
I heard from several people that the silencing of the birds is really remarkable. The mosquito thing is cool.
So many mosquitoes.
Where'd they come from?
There's sort of a swampy area nearby, so I guess from there. But it was sort of like it was getting dark and the mosquitoes were just everywhere.
So when the sun came back, did they disappear?
They did.
And the birds started singing again.
Yeah, right at totality, actually, there were some geese that were quite loud. They seemed to be disturbed by... there were some geese and some dogs that seemed very disturbed right at the moment of totality.
Yeah.
But the frogs were clearly very excited, as were the mosquitoes.
Cool.
Alan, you're next.
Chris writes, TWiV 1105 on the wasp's gall. Here's a funny for the mob. Sammy comes home to find his wife in bed with another man. At court, Sammy's lawyer describes the reason for the divorce being that his wife cheated and had the gall to do so in Sammy's own bed. Sammy interrupts and says, No, the reason isn't gall, but chutzpah, which is because my wife said, stick around, Sammy, you might learn something. Thank you, Chris.
That's just so nasty.
Yes.
If it's true, I don't know if it's true.
No.
You think it's not?
I think it's a joke.
It's a joke.
Here's a funny for the mob. That's a joke. Yeah.
Yes.
Okay, good. You might learn something. Monica writes, I have an amazing STEM girl that practices stitches and plans to become a virologist. At 7, she had already picked her classes at MIT and where she wanted to live. STEM has always been close to heart, including getting more involved. Noah has always advocated for girls and women, including raising funds for different programs. Last year, after attending a few STEM camps, Noah realized just how much educational camps cost and wanted to see more girls in those programs. Noah started a fundraiser for STEM camps and has continued with that this year, too. Below is her fundraiser, and would appreciate it if you could just look at it and maybe leave some words of encouragement. And so there's a GoFund.me, which is a real organization, and there's a picture of Monica, and who wrote the letter, and she writes, raising money to send girls to STEM camps this summer with the most adorable little dog. Oh my gosh. That's great.
That's awesome. I am excited about girls going to STEM camps.
That's an excellent yeah.
So if you wish, you can donate. They have a $5,000 goal. It's very cool. All right, Brianne.
Yes. Bob writes, "Hi all. There is a conversation online about avian flu virus in cow milk and raw milk. I have been drinking raw milk for over a year now for various reasons. The idea online was that there was some type of equivalency between flu transmission via air and flu transmission via drinking milk. How is it possible to get flu by drinking it? Wouldn't the acid in your stomach kill the flu? Even if it didn't kill the flu, how would the flu virus then go from your stomach intestines to become an infection? Are there any studies supporting your position? Thank you, Bob." I have actually seen some things online and some questions about drinking raw milk and influenza. I believe I saw those links through ProMed. And my guess, I don't know for sure, is that perhaps you might inhale some virus while you were drinking the milk. But I don't know for certain.
So this is unpasteurized. That's the problem.
Exactly.
Yes.
It's raw milk, so it's unpasteurized.
Yeah, so if it's pasteurized, then it's not a problem. It's inactivating the virus.
That-
I would be concerned because when you drink, you aerosolize,
Yeah.
right?
Yeah.
So it's not. I wouldn't worry about the virus in your stomach. 'Cause it will
I'd worry about
most-
the aerosolization.
Yeah, the aerosol in your nasopharynx, right?
And I wouldn't actually be too worked up about the avian flu virus in raw milk. I'd be worried about the bacteria in raw milk, but presumably, Bob, you're already well aware of the risk
Yeah.
that you're taking by drinking that.
There are some producers who are very careful. I don't know what they do.
Yeah.
They claim some people believe that they can produce raw milk without the risk of giving everybody listeriosis or E. coli or Campylobacter or Salmonella, and and then you see something in ProMed or on MMWR about how yet there was another outbreak of this from raw milk, which is why it's illegal in a lot of places. So, potentially avian flu virus because of the aerosolization when you're drinking, and also all the other things that we know is in your raw milk.
I don't know how many.
There's a certain irony in being concerned about an infectious disease when you're drinking raw milk. Just going to leave that sitting there.
Yeah.
I think the numbers, though, how many people actually drink raw milk and how many of the cows are infected?
Surprising number of people drink raw milk.
Really?
I have heard of it from a surprising number of people, and as someone who home pasteurizes her own eggs and flour to eat cookie dough, I'm on the other end of the spectrum of eating extra pasteurized food, so I am not drinking raw milk.
No, I don't think I will either. Well, we'll see. I haven't heard of any infections so far, but it's not easy to get infected by this virus. So let's see. Actually, we can think about it. The receptors for H5N1, they're alpha-2,3 linked sialic acids. They're typically down in the lower tract. The only ones in the upper tract are in your eye. There are some alpha-2,3 linked sialic acids in your eye, and there has been at least one outbreak of H5 avian influenza on a poultry farm in the Netherlands where the virus initially infected the eye. And then, as you know, there's a duct that drains the tears to your nose, and then it can go to the rest of the tract that way. But I think it's hard for you to drink milk and get it in your eye, probably.
Unless somebody tells a funny joke and you laugh really, really hard.
And you spray it out. Yeah, that's true. Anyway, there you go. Who's next? Alan? Yeah.
I think so, yeah. Anthony writes, "There have always been lunatics and idiots, few and far between and easy to spot. The scary thing now is the tsunami of the mentally mangled who apparently are functioning." Links I haven't read this, Chicago Tribune story. Trump's Illinois delegates. election denialists. Some claim vaccines were useless, or QR codes led to government tracking. Yeah, so this is the community of people who are lining up on that side of the aisle.
They think they put chips in you along with the vaccine so they can track you. Yeah, it was just ridiculous. Come on, use your head. Just use your heads. Or maybe you don't have the background, but listen to people who can. There have always been lunatics and idiots. A tsunami of the mentally mangled. Oh my gosh. I don't know if they're mentally mangled. They just don't want to think about this.
They have a sense of community. I've talked about this before. There's a sense of community in holding beliefs that are at odds with the mainstream, and they are getting something psychologically from that, and so they are attached to those beliefs for completely non-rational reasons.
Next one's from Martin. Dear TWiVers, I should be grateful if you would kindly consider Professor Angus Dalglish's prompt reply to my inquiry about SARS-CoV-2 vaccine boosters causing cancer to reemerge in some of his patients, after spotting a commentator assert that vaccines can cause this disease on the Daily Telegraph website. I think you should say, after spotting a commentator on the Daily Telegraph website, because it sounds like the disease is on the website. You know what I mean? I'm just picking your grammar. I seem to recall discussion recently in TWiV about suppression in T cells after COVID-19 vaccine booster shots. Has this been noticed widely through the vaccine community since the SARS-CoV-2 virus pandemic? Okay, so apparently Martin wrote a letter to Angus Dalglish, who I've heard of. Probably you have too, Brianne, right?
Mm-hmm.
Is he an HIV/AIDS person?
Yeah.
And Angus Dalglish writes, unfortunately, it's true. The booster does perturbate the immune system, severely leading to T-cell suppression, especially in cancer patients, and IgG1 and 3 switching to IgG4, which is tolerogenic and desired in transplant patients. I've now witnessed 12 patients who were stable with melanoma who relapsed after the booster. 3 of my friends who only had a booster to travel developed bone metastases within days of the booster. 2 are already dead from their turbo cancers in spite of the very best treatments. Having nearly 30 years in developing vaccines in HIV and cancer, I have warned that there are models where 1 and 2 shots are good and the third causes inappropriate immune responses, destroying any benefit from the first 2. I've sent many co-written letters with dozens of colleagues to WIDI, NHS, DOH, MHRA, none of whom will respond. I've written about this with references to published work supporting the evidence in the Daily Skeptic and TCW Defending Freedom. Okay, maybe they're not answering you because it's wacko. Your experience is not data. You know, if someone got melanoma after the booster, it's an association. But I wanted to ask Brianne, first of all, what is this that the boosters perturbate the immune system, leading to T-cell suppression?
I have heard rumors or arguments on this, and I have not seen any data to support it. And so I don't know.
Yeah, I haven't either. And the switching to IgG4, there's no known consequence of that, correct?
I mean, there's no particularly known consequence. There are some differences in antibodies between vaccine-elicited responses with SARS-CoV-2 and with infection-elicited responses in terms of isotypes and, in particular, glycosylation of the antibodies, but nothing that's really clear here or that would be pathologic with the vaccine.
I think Angus should know better about, hearsay. It's unfortunate. And it's when he says turbo cancer, that's just, there's something off there.
Yeah, and Dalglish has taken a number of poorly evidenced stances on a number of issues. He was a prominent medical researcher. I think he co-discovered the CD4 receptor as a receptor for HIV, so he did some important early work. But, just because somebody was a good scientist at one time doesn't mean they continue to be so. And I think if you look at his Wikipedia page, you can get a good idea that he's not necessarily basing much of his opinions these days on a sound basis.
I have, by the way, I have included some letters to Daniel, which weren't read on his program, but I thought are useful here, and that's some of them are coming up. And we're back to Brianne.
Yes. Chris writes, "Hello, Vincent. I just finally had a chance to listen to you and Daniel from Wednesday, March 20th, and feel compelled again to write, likely when you were riding on the 948 home. I come for the science, sure, but gain so much just by hearing you two together. You speak so well and are kind and empathetic, treating every question and person with respect and consideration. It's a big commitment. I'm sure Daniel would rather be sailing. I had a sloop when I was young and fully understand the pleasure of being on the water. People listen to guided meditation and attempt to capture similar good vibes. You two deliver them without even trying. I feel so good listening to you both. I had to write to thank you. I take your science and warmth with me every session. And just a side note that Tim's mirror mirror paper was stunning. Every cell with a membrane has a sense of self. I know, Steph, it is deep, Tim. Many thanks again and fondest regards, Chris, who is in Toronto. And he says, it is sunny and warm with a nice blanket of TWiV and snow.
Thank you. Thank you, Chris. It's very nice. I'm glad you like it. And, you know, things resonate differently with different people, right? That's humanity.
Yeah.
So it's interesting because some people hate us.
They
Or
put
maybe
you on
hate us.
lists on Zitter.
They
Yeah.
put me on lists that I'm a disavower or something like that. Alan, you're next.
Liz writes, I wanted to write and express my heartfelt thanks for the work that you and Dr. Daniel Griffin have done over the past few years. I was listening to the weekly update today and was touched to hear Dr. Griffin speak about POTS, P-O-T-S. This is, what does that stand for again?
Posture orthostatic
Tachycardia syndrome. Right.
Yeah.
His advice on how doctors can screen for this common post-viral issue of the autonomic nervous system meant a lot to me and our family. Having an infectious disease doctor educate other doctors on lingering effects of a virus will hopefully help those suffering receive better care and treatment from physicians out there. It does seem so long ago that I was at home, early 2020, taking care of my college student who had just been diagnosed with POTS by a doctor at Vanderbilt's Autonomic Center. Knowing so little about what caused POTS after a viral illness, I spent time on your website teaching myself virology and trying to better understand what had happened to my once healthy child. I listened regularly to your podcasts about SARS-CoV-2 and recognized immediately how the tail of this virus sounded so similar to what my daughter experienced before COVID-19. It has unfortunately taken a pandemic to push our medical system to recognize and address post-viral syndromes. Hopefully this will lead to better research and treatment options. I hope you can keep us posted on this progress. I'll be listening every Saturday. Thanks again, Liz. Thank you, Liz.
I think it's very interesting that long COVID has, what's the right word? Given some
Galvanized research?
Galvanized research on and back to MECFS as well,
Yeah.
right?
Mhm.
Post-viral syndromes. And, you know, for years, those patients were upset that nobody paid any attention to them. And there's very little money invested by NIH. And that's why they were so excited when XMRV was a potential cause, remember?
Yeah.
Which turned out to be fake. But I think now, with long COVID, everything is much better because people now believe it.
Yeah, it's sort of like we're now realizing that perhaps in some people there's just long virus.
Yeah. Yeah, and it's not, I mean, it's not just that people suddenly believe it. People have had the idea that, yeah, they're post-viral syndromes, but we can't really study them because they happen at different times in different people. And you look at the patient population and it was really, really hard to get a grip on any of it. And now with long COVID, you know, I think we talked about a while ago, it's not exactly a silver lining, but one of the things that really stimulated this is you've got a huge patient population, and you know, in many cases to the day, when they got the virus that led to these subsequent sequelae, and so you can really do the kinds of research that can get at these things.
All right. It's me, right?
Yes. Oh, wait.
Anthony,
Yeah, that was, yeah.
You just did that. Anthony writes. So this is a transcript from one of Daniel's clinical updates. Bus driver says, "Is there any reason why I shouldn't kiss my cat on the mouth?" And Daniel says, "You can kiss your cat in the mouth. It's a relatively safe thing." I remember Daniel said that. Oh my gosh. Why would you want to do it in the first place? But
Don't do that.
gosh. So it's, this one he was on the live stream a couple of weeks ago. That's right. So he gives a reference for cowpox virus infection and orbital cowpox. I don't know why. Maybe they're related. Let's see. An 11-year-old girl presented with a lesion on her cheek. It was cowpox virus. I wonder if they think she got it from a cat. I don't have the full article here. And the other one is...
we covered it. Apparently, we covered it on TWiV 490.
Orbital cowpox, which looks horrible. Oh, and this person got it 2 weeks before symptom onset. Her pet cat had developed lesions of the paws. And then this lady got redness, irritation, and discharge in the right eye. I mean, her eye looks discharged. Oh, my gosh. It's more than discharge.
No.
Ooh, it's inflamed like crazy. So I guess, yeah, you can get cowpox from your cat. And so then Anthony writes, perhaps the definition of relatively is relative.
Yes.
Still, in terms of risk reward, I'd say don't do it. Yeah, I would agree. On a related note, I wonder how many unknown rodent pox viruses there might be. Rodents are undersampled. And so I think there are probably some, there's mousepox virus, right, which is a famous one. But I'm sure there are more.
Don't kiss your pet rat on the mouth either.
Yeah, no.
Don't kiss.
Don't kiss pets on the mouth.
Pets in general.
Yeah.
Even people you don't know, you got to be careful. A lot of viruses out there. Okay, back to Brienne.
Charles writes, "Hello, Twivers and Daniel. I'm not sure if this is a letter to or a letter for TWiV. I have gone down a very dark rabbit hole and would like some help. The dark rabbit hole is the internet's misinformation news sites' comment sections." Oh, never read the comment sections.
No.
Yahoo is the worst that I visit, but even the Washington Post is inhabited with a fluffle of evil rabbits spreading misinformation. Please don't kiss the fluffle of evil rabbits.
No.
Notice I did not say I wanted help getting out of the rabbit hole. I want help countering the misinformation, not from you directly. You do a great job, and I don't know how you have the time to do what you do already. I am looking for help from your students. Not just your students, but from all the students that listen to Microbe.tv. I can be an ambitious fellow. Ideally, a class getting a login on Yahoo, as Team Drew, for example, could check a few sites each day and counter the most offensive rabbits, such as Paul. This misinformation is not just COVID-19 or vaccines. It goes beyond that to things like birth control. See the story in the Washington Post, and he gives us a link to a Washington Post birth control misinformation story. It pisses me off to see so much misinformation go unchallenged. So please recruit a little help so we can be a real PITA to the misinformation spreaders. Thanks, Charles.
I just don't think you can make any inroads.
Yeah, I don't know that you're going to convince people. I mean, maybe you convince the others who are reading the comments, but don't read the comments.
All you're going to do by participating in this, and all you're doing if you're participating in it now, is pouring gasoline on it. And the trolls who are in there posting this stuff, often by copy-pasting it from other arguments that other people have made, they can do that with very, very little effort, and you cannot win. Because the people doing that it's not coming from a lack of information. It's not coming from a misunderstanding that you can correct. It is not a discussion that is open to further input. You need to get out of that hole. Turn comments off by default in your browser, or use a content blocker. And personally, I think news sites, especially credible ones, ought to already have disabled comments for exactly this reason, because nothing good is coming out of it.
Yeah, I often, when I read Paul Offit's Substack. I look at the comments. It's just totally
No.
illogical, right? Totally crazy. So who said you can't argue someone
You
who's
can't
used
reason someone out of a position they didn't reason themselves into.
into?
Yes.
Yeah, that's basically it. It's all nonsense. Okay, Brienne I'm next. Sorry, not Brienne, right? It's me. Hunter writes, TWiV team and particularly Angela. This is hot off the presses and seems to be a bit concerning. So this is an article of the first instance of avian influenza in goats from March 19th. What a cute little goat. I know this is a stock photo. But goats are cute.
Baby goat, yeah.
Baby goats anyway. Adults, Yeah, highly pathogenic avian influenza in goats. Wow, cows and goats. Oh my. I do not see it as an issue for specialized livestock production. My concern is the backyard farm that has multiple species and minimal veterinary interaction. Yes, I would agree with you. And let me tell you a story. I was at the University of New Hampshire. Is that in Durham, New Hampshire?
Yes. I think that's the
And
one
I
that's in Durham.
spoke with a clinical microbiologist, and he said they have a lot of mom and pop farms in that area. And he said they got a call from one of these small farms, and they said our chickens are acting weird, right? And so they drove a couple of hours north, and by the time they got there, they were all dead. And he said they found H5N1 influenza virus in them, and what happened? They had a little pond on their property, and the ducks flying over, they go to drink some water in the pond, and they leave influenza virus behind, and then the chickens drink the water, and boom. So yeah, most big productions, they're inside, right? You have good biosecurity. So yeah, that's a problem. When I asked Ron Fouché back in last year at a meeting in Baltimore, I said, "Are you worried about H5N1?" He said, "No, because it was then just expanding globally more than it had ever had before." He said, "I'm not worried about people, actually. I'm worried about animals, mostly. Like the sea lions that died off the coast of Peru and many others." She said, "I'm worried about that. They're going to get infected and have issues. I'm not worried about people." So,
Did you know that you can actually rent goats to mow your lawn?
no.
Yeah.
Really?
Yes.
Will they do a good job?
Eventually, maybe?
I think so, yeah.
I mean, it would take probably a couple of weeks, right?
I guess it depends on how many goats and how big your lawn is.
Well,
Yeah.
then you're going to have a fence because they're going to just wander
Oh, of
off.
course.
So
This isn't-
my understanding is that goats kind of nibble a little bit here and there and go around. They might eat the rest of your landscaping shrubs.
Perhaps. But apparently you can get goats to come do lawn maintenance. There's also things like goat yoga. There are some people who really find goats cute and really like goats a lot.
Yeah, I saw on Instagram of this tiny goat. It was like in the palm of someone's hand. It was just so cute. Yeah, animals can be very cute. Okay, that was me. No, who read that?
I think I got skipped.
Oh, sorry, Alan.
So I
Go
think,
ahead.
did
Charles.
Brianne read the one about the rabbit hole, or did I?
I did. I read the rabbit hole.
You read the rabbit hole, and then Vincent,
I
we skipped to
skipped.
Vincent for
Okay, so why don't you
Hunter's
go?
letter about goats. Should I pick that up now?
Yeah, sure.
Okay, so Charles writes, "Hello, Twivers. After listening to TWiV 1095, my odd sense of connections put together some of the unrelated thoughts into this odd letter. Comments about wonderful writing, beautiful pictures, and pythons in Florida naturally led me to Beowulf. Adding in that Matters Microbial number 31 was an interview with a University of Kentucky professor sealed the connections. Let me string this together for you. Yes, please. Beowulf is obviously wonderful writing. Over 30 years ago, I had a small part in some of the first high-resolution images taken of Beowulf, meaning the text, not the legendary hero. That work became Dr. Kevin Kiernan's electronic Beowulf project at the University of Kentucky. Today, the project is up to version 4 and has some really beautiful images at much higher resolution than we could do in 1993. The project also has images outside of the visible spectrum. That was something we were not allowed to do 30 years ago, and would not have been able to do a very good job of if allowed. And that is where pythons in Florida come in. About 20 years ago, I was part of a team that put together a multispectral camera system for a USDA water conservation lab that could image light from 400 to 1100 nanometers. 400 to 700 nanometers is the visible range. I no longer have access to such systems. I hope one of your listeners may have access and the desire to go high-tech snake hunting. My thought is that there is very little, if any, evolutionary pressure on pythons to have camouflage that works outside of the visible spectrum. If the snakes could be imaged with a multispectral camera system, it may be possible to find some wavelength or wavelengths that will provide good contrast with the background. That information could then be used to make a better snake-finding camera. Sounds like a good project to me. Come on, University of Florida, Florida Museum, you can do it. And provides a link to the Electronic Beowulf project. Charles Fisher, loyal listener and PITA. Well, thank you, Charles. That is a fascinating idea. So extending the wavelength down into the infrared, I guess, is the idea, and then maybe you could spot the snakes more easily?
Yes, although I have a question for Charles and in his plan. So he mentions little evolutionary pressure to have camouflage outside the visible spectrum. But aren't there other organisms that have a slightly different range in the visible spectrum? So this would be little evolutionary pressure to avoid humans' vision, but what about other organisms' vision?
That was that occurred to me as well. Like, what is the evolution? What is the spectrum of common raptor species or other predators that might prey on small pythons? And nothing preys on big pythons. That's part of the problem. But yeah,
The Electronic Beowulf project. Very interesting.
Very cool.
Okay, let's go back to Brianne.
All right. Brenda writes, "Hi, Vincent. Just thought I would draw this to your attention." And she gives a link to a UK article that says, "COVID-19 lab-like most likely cause of the global pandemic, new study shows." Boo. Can't say that I am terribly impressed about the validity of a conclusion reached by university scientists in Australia using a technique I have never heard of on data we do not know the veracity of, which is full of unverified allegations. Don't know who would be the best to question it. Debunk the funk, Beyond the Noise, or somebody else. And this is from Brenda from Black Island, Scotland.
So this is all wrong, as usual. I mean, they say that the Wuhan Institute of Virology was only 8 miles from the Huanan seafood wholesale market. Really? Well, where was the early epicenter? This is one of the scientists, Dr. Chen, in Australia. Where was the epicenter? It was at the market. It wasn't at the Wuhan Institute of Virology. If it were from the lab, it would have the epicenter there. It's just nonsense. It's all nonsense. And they say, oh, the lab had this, they were researching this virus. It's RATG13, which they don't say, but it's 96% homology with SARS-CoV-2, something that was revealed only after the pandemic began. It's not true. It was revealed before the pandemic. And 96% is 1,200 bases away. Can't be the ancestor of SARS-CoV-2. This is all nonsense. It's just crap. The same crap wash, rinse, and repeat, you know?
Yeah, so the tool that they used for this, I'm just looking it up. It's the Grunow-Fink Epidemiological Assessment Tool. There is established science on this already from 2017, where they applied this. It's supposed to be it's an analytical method to try and determine natural from unnatural outbreaks. And so this group, Chen, Chugtai, and McIntyre, this is from Military Medicine, 2017. They did a systematic review where I think they fed this and other assessment tools data on various known epidemics, and they found that the Grunow-Fink tool was particularly bad at identifying natural versus unnatural. So this seems like it might have been kind of a setup.
Yeah, if you were going to have such a tool and you were going to train such a tool, wouldn't you need to have a lot of definite evidence for unnatural epidemics?
There are, right. So
you could train that tool?
You could put in things like the Sverdlovsk incident and a few other known lab-based outbreaks, but the data set that you would train this on would be necessarily small. And so it's not terribly surprising that these are not the most reliable tools. But I would just happen to find that there's, in a quick PubMed search, a paper that actually points out that this particular tool is not very good.
Also, I think you should put into the equation the fact that every human virus comes from some animal. I mean,
Right.
I'm sure they don't bother to do that.
Well,
Right.
whatever this tool does, I think the more important thing to note is coming out and saying that this proves that it was most likely due to a lab leak. It's no. If you're using any kind of analytical model, even if it's a reliable one, which this isn't, then you would say, this model predicts that it might have.
Yeah.
You don't come out and say this is not any kind of proof.
No, and there's good evidence for this having natural origin.
There's a
related
lot
to
that's
animals in the
very, very hard to explain away if you think that it came from a lab source.
Yeah.
Also, it's the same arguments used over and over again.
Yeah.
And there's no coherent narrative as there is for a natural origin. There's a very coherent narrative. The lab leakers take pieces of this here and there, and it's just the same stuff over and over. Nobody's going to deal with this, Brenda, because it's more of the same. There's nothing new.
The only coherent narrative is that people want something to blame or someone to blame.
That's right.
Yes.
That's right.
That is the attraction of this idea.
We will probably never know where it came from exactly, and people don't like that, so they come up with something else. Okay, let's reboot here.
So now it's my turn.
Alan, you go, please. Thank you.
Who read the last letter? All right. Robert writes regarding TWiV 1094 and 1092. Number one, recommended ages for MMR. Children are typically recommended to receive a first dose of MMR at their first birthday after maternal antibody has waned, and a second dose before starting kindergarten at ages 4 to 6. However, children are recommended to receive doses sooner if traveling abroad, as many cases in the U.S. have occurred in young children who were exposed overseas. CDC recommends that all U.S. residents older than 6 months of age who are planning to travel internationally receive MMR vaccine prior to departure. And gives a list of the specifications there. 1 dose before departure. Children 12 or older should receive 2 doses separated by 28 days, or 12 months of age or older. And teenagers and adults without evidence of measles immunity should have 2 doses of MMR. Protect your kids before those spring break summer trips abroad. Recent COVID-19 vaccine efficacy data were presented at the ACIP Advisory Council on Immunization Practices meeting in February, contrary to comments in TWiV. Okay. I don't know what we got wrong there.
So this is all from Daniel.
Okay.
So if I'm going to Europe, I should get an MMR vaccine? Is that what CDC is saying?
I was trying to figure that out myself because it says teenagers and adults without evidence of measles immunity. So does that mean because I can tell I have my vaccine records and know when I got measles shots, does that mean evidence of measles immunity, or should I get it titer?
No, I think, well, you could. Daniel said you can get it titer, and they know the neutralization level, that's the cutoff. But yeah, I think this is for people who are not sure. If you've not been fully vaccinated or have not had measles, and you go places where measles is spreading. So it's not everyone.
Yeah.
I did get measles vaccine, but that was a long time ago. And I just don't know that it's a good public health thing to have to do a titer on everybody, right?
No, that's too much to ask. But if you aren't sure whether you were vaccinated or had it, and you're gonna, then you should probably get the vaccine. And he finishes up. Dr. Ruth Link-Gellis reviewed preliminary vaccine efficacy data on the updated 2023 to 2024 COVID-19 vaccine, including the Vision and IV data published the next day in MMWRs. Gives a citation there. Thanks to TWiV and your ongoing dedication. And Robert is doctor somewhere.
Volker writes, thanks so much to Alan for stating clearly on epitope 1091 that we cannot simply switch to IPV worldwide. So often during the last months, when especially you, Vincent, repeat over and over again that NOPV was a failure and we should not use a vaccine that paralyzes kids, I was shouting in my head, don't neglect money, don't neglect reality. Wow, it's a good trade-off, huh? Finally, Alan formulated it better than I could have done. Of course, in an ideal world, we would only use the best vaccines for everybody. In this world, there would be no hunger, no war, no resource scarcity, no climate change. This is not our world. In our world, we have to distribute our resources, and if this means that the money necessary for all IPV distribution and infrastructure can provide more good for other things, then we should stick to NOPV for now. Greetings from Bavaria. I don't agree. I'm sorry. I don't want kids paralyzed by a vaccine. It makes things worse, not just for them, but for vaccines in general. Gives them a bad rap. So I think I agree that the perfect world is not attainable, but some things we have to fix.
I think the lesser evil is to go ahead and give them this imperfect vaccine.
You think so, huh?
Yeah, and public health, and especially public health in poor countries, is full of choosing between bad options. This is another one.
Okay. We can
But
disagree.
I do, yeah, we can agree to disagree. I do see your point. I understand, it does a lot of damage to, to the kids who are paralyzed, but also to efforts to vaccinate when you have a vaccine that can cause the very disease it's supposed to prevent.
So we can disagree, and we will not call each other
We will not
names.
put each other on lists of bad people, no.
Yes, this is the way it should be done. Okay, back to Brienne.
All right. Craig writes, teratoma today, TWiV 1095, prompted me to complete an email I started drafting some time ago. Teratoma is a good word from a good family. Teret, from Greek, plural of, means a marvel, a prodigy, a monster. I'm sorry, I cannot pronounce the Greek. See teratogenic, teratold, etc. So whenever you think terat, think monster. Tera just means big, specifically the prefix 10 to the 12th from the same root. Alan Dove lives in Massachusetts, which means he's eligible for a Boston Public Library e-card, and he gives a link, which gives one online resources, including the Oxford English Dictionary, and we have a link, the authority for meaning and etymology of every English word ever deemed significant, greater than 600,000 words. I find the etymology or history of technical words to be very useful. Some are directly meaningful in understanding, like teratoma or macrophage. Others historical, like eosinophil, and some counterproductive. But we're stuck with. Complement is a common word, but when you see it in immunology, it's not obvious what it's complementing. And is it complement, L-E-M-E-N-T, or is it complement, L-I-M-E-N-T, and it's saying your immune system is very nice. I can actually tell you what it's complementing. So a suggestion or something to consider. When on the podcast and in your lectures, you introduce a term, break down the etymology and meaning. I find this helpful in understanding and remembering. Bernard Sue, Farm D, aka Chubby Emu, does this in his videos, and while it becomes a running joke, it does seem useful. Gives an example and a link, where he breaks down the 4 parts of acute hyponatremia. I see that the star fill cells are named by what stains them, which is helpful to remember. And it's fun to know that the phag in phagocytosis is the phag in sarcophagus. For the students that probably have encountered these terms before and may have learned the meaning origins, but a reminder doesn't hurt. You could also make a page with your favorite technical terms, like enterovirus, which was mentioned by another letter writer, and link to that in the comments for each episode. End suggestion, begin general comments. I followed Professor Racaniello's 2023 virology lectures, and those are very interesting, but that led me to Professor Barker's immunology lectures, and those are really interesting and useful. I think people should have an understanding of the science behind the things they use. Enough thermodynamics to use a microwave oven well, enough mechanics to do things around the house, enough about cars to know how to fix simple things and work well with the mechanic. I use my immune system every day, more than I use my car. I see that immunology is like thermodynamics. It's not a linear story. Material taught earlier really only makes sense when you understand the later material. I only felt I understand thermodynamics after 2 levels of classes and then reviewing for quals and teaching it myself. I don't plan on taking high-level immunology courses, but I may go through another round of lectures. I'm just getting the high-level general ideas. Things in virology and immunology are changing often enough that I don't think it's useful for me to memorize any details. Phrases like "we used to teach" and "10 years ago we thought" keep coming up in both courses. Thanks, Craig.
I don't think I can stop in the middle of my lecture and give the etymology of every word.
I definitely
No.
can't give the etymology of every word. Sometimes I will give a little bit of background
Yeah.
if it's useful. Complement was originally described as something you could add to antibodies to make the antibodies work better. So it complemented the
Complemented
antibodies.
their activity.
Yes.
All right, Alan, you're next.
Sure. David writes, I don't know if you've discussed this topic before. If so, I'd appreciate a reply pointing to a particular TWiV episode so I can review it. Listened to Paul Offit on NPR yesterday regarding measles. He made the point that measles/smallpox have long incubation periods, unlike COVID, which is why MMR vaccine protects against mild as well as severe measles disease. I have watched TWiVs, albeit not all since 2020. Don't ever recall hearing about this important distinction regarding vaccine efficacy for mild disease. If you haven't communicated this before, I've even got a title for a future episode on that subject, The Long and Short of It. What a great job you're doing. Our family are all patients of Dr. Griffin's practice as a result, even though residing in Florida and Maine. Thank you, Vincent. Keep up the good work.
Now, when I heard Paul Offit say that, hadn't thought of that before. It makes sense. If
Yeah.
the incubation period is longer before symptoms, which is what the definition is, then you have more time for a memory response.
Yeah.
Mm-hmm.
And, a short couple of days, it's harder. Yeah. It was a good point. Yes.
Yeah.
Carrie writes, I love TWiV. I listened to every episode during COVID lockdown as I was part of my local health department epidemiology team. I haven't kept up with every episode since, but I have a question regarding rebound COVID after treatment with Paxlovid, which by the way, doesn't exist. There's no Paxlovid rebound, Carrie.
It's COVID rebound.
It's COVID rebound. I escaped COVID infection for 4 years, but finally got me recently. I obtained a prescription of Paxlovid, took it second day of symptoms, completed the medication, felt great, tested day 9 after first symptoms, tested negative. 2 days after this, so day 11 after first symptoms, I had a return of symptoms, runny nose, congestion, cough. Symptoms were less than initially, but I was surprised and tested again, and this time positive. I did a little research, found out that there can be a phenomenon called COVID rebound after finishing a round of Paxlovid. I assume this is what happened with me. Can you explain the virology of what happens during rebound? I'm curious to know why there's a rebound. What does it suggest about treatment with Paxlovid? If you have already discussed this, direct me to the podcast. We have on Daniel's podcast, but we can repeat it.
And she is phrasing it correctly. Rebound COVID after treatment with Paxlovid.
So the natural course of COVID is 2 phases roughly. There's a first phase, about a week or so, the viral phase, where virus is introduced in you, it replicates to a peak, you get the symptoms in the upper respiratory tract, and then the infection is cleared. And then in many individuals, you have a second phase, the inflammatory phase, where now the immune system is sometimes overreacting, and all those symptoms are immunopathology, and that can cause severe COVID in many people. So when people go in the hospital because they can't breathe, it's typically in that second phase. And antivirals will not help you in the second phase because the viral titers are already low. So that's what rebound is. It's not really rebound. It's the second inflammatory phase. It will happen whether or not you take Paxlovid. And the testing positive on the antigen tests, so Daniel and I have talked about this, there's probably residual antigen in different parts of your respiratory tract that are potentially sampled when you do another rapid antigen test. But there's no evidence that rebound involves a risk of new transmission. In other words, there's no increase in the production of infectious virus. It's just a symptom. So you did the right thing. You took Paxlovid. It made you well. Okay. Does that cover it all, folks?
I think so.
Yeah.
Back to Brienne.
I'm laughing as I read this, as I see this letter, because A, it's going to result in me having a quote somewhat like lungs are an important organ. But also I feel like this person actually may have been in my lecture, my microbiology lecture this morning, as there was a point that I kind of ranted about a little bit in that lecture. I have no expertise in sloths. This allows me to freely engage in speculation and theories about their relationship to algae. A thought on your question of whether algae and sloths are symbiotic. I am sure it is useful to the sloth to have algae on it to improve its camouflage. Anything that adds complexity to the shape boundaries of an animal makes it harder for a predator to see. Think parrot colors for an extreme example. For the algae, free transport is a core benefit. I have never been sure what the bar is for symbiotic. Does the sloth benefit sufficiently from the algae is hard to assess. So
Didn't you say to us that sloths were green, Brienne? Was that you?
I have not been a part of any previous sloth discussions on TWiV, though I, like Jerry, have no expertise in sloths. But I did actually teach some things about
I have
symbiosis.
quite a bit of expertise in sloth.
Yes, I
but
do
not
know quite
in
a bit
sloths.
about sloth. I did, however, discuss symbiosis this morning. And symbiosis is really about an interaction between two organisms. And there can be an interaction where one organism has a benefit and the other does not. A sort of +0 interaction, that's commensalism. So as long as one party is benefiting, it is a symbiosis. I would assume, if nothing else, the algae is getting a stable environment and perhaps some nutrients from the sloth. So it is at least a commensal symbiotic interaction. It may be a mutualistic if the sloth also receives a benefit. And I don't know enough about sloths with my lack of sloth expertise to address that. But I may have also gone on a rant in microbiology this morning about why certain microbes in the GI tract are called commensals when it's really a mutualistic interaction.
Yeah, well, it's all about terminology, right? Yeah. Someone on TWiV must have said that about sloths having algae on them because they move so slowly.
Yeah, I don't remember this.
I don't know who it was. Alan, you're next.
Okay, Yosef writes, "Hi all, my lab delivers both lentiviral and AAV preps to cells in vitro. The titers for lentis come as transducing units per mil, whereas AAVs are in genomic copies per mil. At a functional level, it appears as though one lentivirus TU is the equivalent of greater than 1,000 genomic copies per mil. Do you know why this is the case and why they are reported differently? I'm sure it's a qPCR quantification as opposed to a strictly cellular one, but I'm not entirely clear on this. At the ground level, by these numbers, a functional multiplicity of infection for a lentivirus may be, say, 3, whereas for AAV it can be 10,000. This is very confusing and could use a TWiV explainer. Thanks all and take care." So this is sort of a PFU, particle versus PFU type of discussion,
I've measured
right?
both of these things.
Okay, take it away, Brianne.
So one measures lentiviral transducing units by actually looking for transduction of a cell, as he said. Oftentimes when I do it, I'm using a GFP lentivirus, and so I look at how many cells become transduced with GFP, become GFP positive after that. And I have some specific math I do to come up with transducing units per mil based on the amount of virus I used and the number of cells I used and that kind of stuff. Whereas AAV, we do entirely by qPCR. And so we are looking at how many genome copies. We don't know how, if those are encapsulated or are going to be infectious. We just know how many copies of the nucleic acid are present. And when I first did this for AAV, I actually asked a lot of questions about that exact thing. But the reference units for AAV are all everyone using the same qPCR primers for looking at genome copies for AAV.
And that is presumably because you can't easily do an infectious quantitation for it?
That is my understanding, yes.
Right.
And,
so this is basically a chemical assay versus a biological assay.
So the problem with the PCR is you're amplifying a little piece of the genome, so they're probably DNA fragments broken.
Oh, absolutely.
Right?
Absolutely.
So that's why you have to add more.
Right.
Because you're overestimating the number. It shouldn't be really genome copies because it's not a genome that you're measuring.
Right. You don't know how many of those are, going to
Whereas
actually be infectious.
The lentivirus is a functional assay, and therefore you're looking at viruses that deliver whatever gene it is to the cell, and so that's why the MOI is lower. Yeah. So hopefully that helps you, Yosef. Alan, you're next.
That was me.
Great.
So you're next, Vincent.
You can tell we haven't done an email episode
we haven't done an
all
email episode in a while.
Anne writes, I was wondering if you had an opinion on this interview with Scholtold David, in which he claims he regularly finds errors in peer-reviewed scientific papers. I was hoping that peer review catches stuff like this, but according to him, it does not. Is he right? Is he alarmist? Oh, yeah. So this story came up some time ago. It's the New York Times. He hunts sloppy scientists. He's finding lots of prey. If researchers aren't getting things right, what else might be wrong? So this is a guy who just pours through paper and looks for mistakes. And so I don't think that you should condemn the entire scientific process by this. Peer review isn't perfect, and a lot of errors make it through peer review. And in fact, maybe some things are wrong, but if something makes a difference, if it matters, somebody else is going to try and do it, and it'll get fixed. That's how science works. It doesn't work on the basis of Alan Dove's one paper, all right?
Thankfully.
It works on Alan Dove publishes an observation, and if anyone cares or if it's important for something, people will do it, and we'll find out the answer. So, yeah, you're going to find errors. You're going to find mistakes all over the place because it just happens and people are really busy. But overall, the system works.
Yeah, it's really about the preponderance of the evidence, not one individual piece of evidence.
And the reproducibility of it. So if I publish a paper, well, back in the day when I did, and then somebody looks at it and says, oh, wow, if that's true, then this other thing ought to be true. And they would then try to replicate what I did in order to extend those results. And if they find that that doesn't work, then, oh, wow, Alan screwed up. And so the process is self-correcting.
Okay, back to the top. Brianne.
Laura writes, hi, folks. I asked this on the YouTube channel, but I thought I'd shoot you an email here also. Here's the question. How likely is it that someone can get infected with 2 strains of COVID-19? And if that did happen, would it make a difference in the course of their disease? My housemate is positive for COVID, and 2 of his good friends are also positive. I suggested that he could go to hang out with them at their house, but he's worried about spreading a second strain. I Googled for this in the past TWiVs, since it seemed like a question you would have answered, but I didn't find any info. Thanks so much for being excellent humans and for all that you do for science education. Laura from Albany, Oregon. A quick weather update. Fortunately, it is 45 degrees Fahrenheit, 7 Celsius here, and not 19 like it was last week.
I don't see why not, right?
Yeah, I don't know any specific examples of superinfection with SARS-CoV-2, but I don't know why not.
On the other hand, if your friend got it and somebody who lives nearby also got it, the odds are that they probably have the same strain, but maybe not.
So if you're in a place with a bunch of people and someone came from another geographic area where another strain is circulating, it's possible. Depends where in your infection you are, because you're going to have, after a few days, you're going to have some interferon, which is probably going to, resist the second infection. But I think we all get multiply infected all the time. So, yeah, we don't have examples of this, do we? I seem to recall some papers where they're sequencing isolates from people and they say, oh, this person had 2 variants in it, but I don't remember where that was. So I think we've seen it. Maybe it was another virus, but
I kind of
yeah,
wonder if those were some of the long-term infections in
maybe,
immunocompromised people.
Right.
maybe. But sure, why not? But I don't think you should worry about it. I think if you're already infected, wear a mask or stay home, right? Alan?
Burnt writes, Dear Twivers, thank you for carrying the torch of science. I discovered your show during the start of the COVID-19 epidemic thanks to a former postdoc of mine. I've been a regular ever since. I have a question and a comment. They are entirely unrelated. First, a couple of weeks ago, you covered Cafeteria ronbergensis. You were speculating about the origin of the species' odd name. The species was described by Fenchel and Patterson in 1988. Tom Fenchel was a professor of ecology at Aarhus University. Part of his work would have taken place at the university's field station in a tiny village named Ronberg. In Ronberg, there was a diner named Ronberg Cafeteria. And sends a picture. And there it is. When I was a PhD student at Aarhus University in the department where Professor Fenchel had been, I was told that he, in fact, had named Cafeteria ronbergensis after this diner.
That's great.
I like it.
Nice.
I think that's beautiful.
Yeah, they should put that on the menu.
By the way, I visited this university and I was told how to pronounce it properly. It's Aarhus.
Aarhus.
Oh, that was not what I was going to go
I would
with.
not have guessed that.
No, neither did I, but they correct you when you say things wrong.
Okay. Aarhus University. I'll try to remember that. Second, occasionally the claim is made, including, I believe, on TWiV, that there is a minimum number, greater than 1, of virus particles required to start an infection. Why would that be so? Should one imagine some kind of synergy, say, to overwhelm local defenses? Or should one imagine that a minimum number is, as the substantial fraction of virus particles are somehow defective, unable to initiate an infection? Best wishes, Burnt. For a lot of viruses, this gets back to what I thought a previous question was going to be about and was a little different. There are actually a lot of defective particles. And so you need more than one to be sure that one of them is actually going to initiate a productive infection. Also, depending on the route of infection, there could be survival issues where, if it's a digestive virus and it has to survive through the stomach, odds may be slim, and so you need a critical mass of virus going in so that something will actually survive. That doesn't mean you couldn't get it from a single infectious particle. It just means you're really unlikely to. You're going to need a bigger inoculum.
So you could imagine mucosal surfaces. Your cells are covered with mucus. The virus gets stuck in it.
Yep.
Can't infect. You need more to try and overcome that. And then once you get into cells, there are interferon responses. So, yeah, all of that plays a role.
The odds are against it initially.
The odds are against viruses. That's why they make so many progeny.
Yes.
Howard writes, as a yearly donor through Parasites Without Borders, I urge you to expand your fundraising efforts and your campaign for a citizenry that is better informed about how science works beyond Microbe TV's audience. Being located in New York gives you easy access to any number of shows with a large reach into the general population, all of whom are looking to book interesting guests. Some that come to mind are CNN's Anderson Cooper, MSNBC's Alex Wagner, and CBS's Stephen Colbert. Vincent would make an engaging guest with an interesting story to tell. You all do important work. Microbe TV deserves, and democracy needs it to have a broader reach. Please don't be shy. I don't think any of those people would find anything interesting about my story, but I think I agree with you. I would like to get a broader appreciation of what we do. And, we're trying to grow here at Microbe TV. That's why we need your support. And I know you already support us, Howard, but you can't just cold call Anderson Cooper and say, have Vincent on. It doesn't work.
So Anderson, Alex, Stephen, or your people, whoever's listening, give Vincent a call. Have him on.
Once Dixon was on Stephen Colbert's show.
Yes.
Yes.
He published this book on the vertical farm. And some of Colbert's people caught it and said, hey, this would be cool. Guy's in New York. Let's get him in. And so he was on the show. Did it do much for his sales? Probably not, I don't know what it did.
It meant that when a friend of mine here at Drew had Dixon in her class as a guest speaker, she showed a video of it in advance of him coming.
Okay.
Good.
Okay, back. We just have a few left. I think we can get through them, Brienne.
All right. Howard writes, "Hi, Twivers, another great episode. The discussion about human, swine, bird viral exchange raised a question that you might have some insight into. Slaughterhouses were a prime location, along with prisons, for SARS-CoV-2 cases at the height of the pandemic, but the reports didn't differentiate between pig, cattle, and chicken slaughterhouses. Do you know if there were any differences between these types of facilities, and whether these differences might have been related to viral transmission? And are slaughterhouses loci for human flu infections, with cattle lower than pigs or chickens? Thanks for your effort. My 100-year-old, 5-time vaccinated mother is in her second bout with COVID in the past 10 months. TWiV's information has allowed my sisters and I to help her navigate her care and all of us to stay calm. Howard in Toronto, where it is sunny and -7 degrees Celsius."
The weather in Toronto has changed as we've gone through these emails.
Yes, yes.
I don't know about pigs, cattle, and chicken. Do you know, any of you?
I don't. I think when we talked about slaughterhouses, we were talking about, the cold air and then not having circulation and not having ventilation as being part of it. I
And
don't
having
remember anything
people-
about the animals.
Yeah, and having people work very close together on a line that's moving very fast, and infection control protocols were probably not a top priority early in the pandemic. So it's really, I think that the spread in slaughterhouses was a lot more about the people than the animals. I don't think it was a lot of spillover events. I would point out
Yes.
that what we see with avian flu is the cases, since it doesn't transmit that well person to person, are often people who work with poultry, not so much in a slaughterhouse, but maybe in a like chicken feeding facility.
Yeah.
So that can make a difference. But I don't think anybody looked closely at whether there were differences based on the species being slaughtered.
Mm-hmm. Right, you're right. It's all about the person-to-person transmission.
Yeah.
You are next.
I think I'm next. Craig writes, "Immune dysregulation. When you next use this phrase, can you please pause to explain it? I've seen people see immune dysregulation or immune dysfunction and mention of T cells in connection to COVID-19 SARS-CoV-2 and jump to saying that this means COVID is like AIDS." I'm sorry to hear that, Craig. This is a case of a little learning is a dangerous thing. Yes, then since you need to know enough to have heard of T cells and understand the prefix dys, but not enough to know what it all really means. I myself only know enough more than that to know that I don't know and would need to look it up and study more before saying anything myself. Thanks, Craig. Yeah, I mean, immune dysregulation just means something has gone wrong in the immune system. So immune dysregulation does not mean it's like AIDS. It could be like lupus. It could be like an ordinary viral infection that gives you a severe inflammatory response. It could be like a lot of things. It means your immune system is
Could be like
doing
what happens
stuff.
if I go out and see that pollen outside.
Yes, that is, yeah. Asthma is immune dysregulation.
So, Brian, what are they talking about with T cells in COVID?
So your T cells may not be making ideal responses, perhaps because you are having too much of an inflammatory response. You mentioned the inflammatory phase, and so T cells are going to respond to that inflammation. And so if you have too much, a problem with regulating inflammation, that's going to eventually lead to T cells being different than making some different responses than they would have otherwise. I think that that's really the big part of this.
And also in older people, that could also be an issue, right?
Of course.
Okay. Chris writes, on the last clinical update, you and Vincent, this is referring to Daniel, talked about how there will always be a new variant every winter. I accept, as you often state, that it is not necessarily more pathogenic. It is not accurate to say that each is more transmissible but more fit. Here's my question. Why is there a new variant every winter? Why is the mutation rate of CoV-2 so high compared to other viruses? On an episode of TWiV a few months ago, I believe Trevor Bedford said it appeared to be more than twice the mutation rate of flu. I recall there being speculation that this was a feature of limited immunity. But surely that's no longer the case. What is it about the features of this virus compared to other viruses? please feel free to correct the premise of my question if I am wrong on any of this, or to redirect it to the main TWiV podcast. I'm just trying to understand how this works. No, it's a good question. So the thing is that if you look at the emergence of variants, that's way beyond the actual mutation rate of the polymerase. So the polymerase, when it copies RNA, it makes mistakes. That's the basic mutation rate. For SARS-CoV-2, it's about 10 times less than most other RNA viruses because it has error correction machinery. But it still makes a lot of mistakes. But why did Trevor Bedford say this? Because the variants are emerging. So amino acid changes seem to be appearing in the spike more rapidly than in the hemagglutinin of influenza
and also
virus.
getting fixed and being observed.
Yeah. So they must confer some fitness to the virus. What it is, we don't know. And it could be that, well, the common cold coronas, which have been around for many years, undergo variation, but only every few years. So maybe with time we will see less of this as the population becomes uniformly immune and so forth. So I think it's a function of immunity, which provides the selection.
Yeah, I think that you should remember, Chris, that we're not just seeing every mutation. When we're talking about variants, we're seeing the ones that were able to reproduce and sort of amplify among the population that had some kind of selective advantage, as Vincent mentioned. And so right now, the mutation rate that being 10 times that of flu is sort of the mutation rate of generation of mutations that have a selective advantage is 10 times higher, not the actual rate of mistakes.
And it's possible that the common cold coronaviruses have already explored the territory that they can cover in their mutation rate and still be transmissible viruses. And so now we see less variation because they're playing variations on a theme, if you will, and they've kind of stabilized in a general vicinity. SARS-CoV-2, still being relatively new to the human population, is still exploring new territory.
So the underlying mutation rate is the same. So the common cold coronas, whenever they spilled over compared to now, the mutation rate is the same. What is different is the variants that are selected, which is not really correct to say that that's the mutation rate. Should be another word for it because it's conflating the two things.
Sort of like the variation in phenotype rate or something.
It's really the phenotypic variation, right? All right, the last one, Brianne.
All right, Portia writes, "Hi, Vincent and the TWiV doctors. Just finished episode 1095. It was a good surprise to hear the pick from Rich is Mark Smith Photography. I have followed his work on Instagram and YouTube since before the pandemic and have even taken 2 workshops with him on bald eagles in Washington state. I can't quite bring myself to go to Florida. He is really an exceptional bird photographer. I am sure he will get a TWiV bump. I will have to tell him about TWiV. As for Angela's new podcast, we are eagerly waiting. One topic I am interested in is exercise, which could build bone mass to prevent osteoporosis for older women. And my husband is interested in the mammalian dive reflex. Thank you and all the esteemed members of your team for bringing all the Microbe TV podcasts. Portia."
So Portia is also a photographer. I follow her on Instagram.
Ah, cool.
Okay, it's time we got all through the letters. That's great.
That's awesome.
Wonderful. Time for some picks. And Brianne, what do you have?
I am exceptionally happy about my pick. So I was looking for something else and stumbled across this article that is called "Cats Are Perfect. An Evolutionary Biologist Explains Why." And this talks about, in fact, in some ways, the phenotypic variation of cats, just like we were talking about with a recent letter, and how, in fact, cats have basically found the perfect phenotype and don't have a lot of variation. There's big cats and small cats, but otherwise they're pretty much exactly the same because they have evolved to fit their niche so perfectly. And this evolutionary biologist kind of talks about how we don't see this in other animals and sort of phrases it like, well, yeah, it's because they're not very good. And so they have to try different strategies to find something that works. Cats found the perfect thing. And so they haven't had to change. You just have a big cat or a small cat. And given my affinity for cats, and I know many of the TWiV listeners' affinity for cats in general and the cats of TWiV, I thought everyone would like to hear about why cats are perfect.
This is
It's so
excellent.
funny. That's just so funny.
But you learned some good stuff about evolution in this article.
Indeed.
Yeah,
indeed.
yeah, it's really a good article.
And then there's links to other cat articles if you'd like,
Yes.
like Why Do Cats Purr, etc. Thank you. That's pretty funny. Alan, what do you have for us?
I have something I stumbled across. I don't even remember how I stumbled across this, but I'm linking to a particular artist who goes by the name Hildegard von Blingen, who does a particular style of music that has been called bardcore. And there are a whole bunch of musicians doing this. She does a particularly good job at it. So she takes famous songs. The one that I've linked to is Paint It Black. And it is a medieval cover of it using instruments from the, 12th, 13th century and adjusting the lyrics a little bit to be appropriate for the time period. And it is just brilliant.
I cannot wait to listen to that.
It is so fun. She's got her whole playlist, and then YouTube's algorithm will toss up more bardcore if you're interested in it.
That's cool.
Yeah.
All right. Mine is a very simple one. It is a quote from Georgia O'Keeffe. I think it's so foolish for people to want to be happy. Happy is so momentary. You're happy for an instant, and then you start thinking again. Interest is the most important thing in life. Happiness is temporary, but interest is continuous. So this really piqued me. It's hard to be happy all the time, right? So, Georgia O'Keeffe was a unique individual, right? And perhaps her thinking is not mainstream, but just wanted to put this out there for people to think about. Because I'm interested in viruses and science, and I've had a continuous interest in that for many years. I wouldn't call it happiness, right? It's interest. She's right.
Yeah.
I'm happy when I have a good meal, but then as soon as I'm done, that's it. The happiness is
fleeting. Yes.
It is fleeting, right? You guys agree?
No, I do agree. I feel like in some ways it's because one of the times when I get really happy is when I'm thinking about something I'm interested in.
Yeah.
And so if you cultivate the interest, then you can, get repeated hits of the happy.
Yeah.
Yeah. Curiosity and wonder are that's better than just happiness.
No, like some of these emails today where we learned things. It's wow, that's cool. It makes
fascinating.
Fascinating, right? And this is from a Instagram account called The Daily Philosopher. Daily Philosopher. It's really good. They have great quotes from all kinds of interesting people. There are a whole bunch of others. Let's see. There was one here. I like this one from Socrates. I cannot teach anybody anything. I can only make them think. Kind of good, right?
Mm-hmm.
Yeah.
What is another one? Here's another one. When dealing with difficult people, remember you're not dealing with creatures of logic, but creatures of emotion. When we are tired, we are attacked by ideas we conquered long ago. Fear is a natural reaction to moving closer to the truth. Anyway, every day, you get one of these, and I really like it. I think it's really good.
Cool.
So that's my pick. We also have 2 listener picks. Julia writes, thanks for the fantastic episodes. I want to share a pick of the week which represents recent success in troubleshooting autoclave challenges in our field research. I'm an environmental health scientist working in Mozambique to characterize enteric pathogen exposures for children under 24 months of age among urban low-income households in Maputo, Mozambique. We collect a variety of matched environmental surface swabs, drinking water, solid and liquid foods, hand rinses, and clinical specimens, stool, blood, which we test by multiplex PCR methods to understand pathogen transmission dynamics in this setting and how child age and hand hygiene behaviors may mediate exposures. The site autoclave has not been working for several months, posing challenges particularly for our hand rinse concentration and downstream processing step. Attached is a picture of a makeshift autoclave using a domestic pressure cooker which successfully sterilized our centrifuge bottles. Yay, feeling quite pleased with this troubleshooting and glad we have a temporary fix to move forward. So this is from Julia, a.k.a. Dr. MacGyver. And it's a pressure cooker. It's great. So you can do that. She is in the London School of Hygiene and Tropical Medicine. And then we have a pic from Charmaine, who writes, Viruses, myelin, the evolution of complex life, and the Fermi paradox. And this is a YouTube video, an exploration of a new solution to the Fermi paradox involving retroviruses and the intricate needs to evolve the myelin sheath of nerve axons that allow for more intelligent life. So I haven't looked at this, but
yeah,
check it out.
I'm going to definitely look at this later because I know the Fermi paradox and find it kind of interesting. And I'm trying to imagine it. I'm having trouble imagining how to put all of these things together, but they're all really interesting things. So I'm ready to watch
I
them.
think the gist of it is along the lines that evolving intelligent life is so complicated that it couldn't have been a common event.
Okay.
Which
it's
I don't
saying
think that really solves the Fermi paradox, but it may be.
It addresses the Fermi paradox.
It addresses
by
yes.
saying that one of the variables is really, really rare.
Yes, as far as we know.
Yes,
but
exactly.
As always comes up in these discussions, we're reasoning from an N of 1.
Mm-hmm.
Yeah. All right, that's TWiV 1107, 1107. You can find the show notes at microbe.tv/TWiV. Questions or comments, pics, send them to us. We like to get them. TWiV@microbe.tv. If you enjoy our work, please support us. It's a lot of fun. This is good stuff. It's science. We're trying to promote it. So please help us to spread the word. If you help us financially, we can perhaps spread the word better and let more people know, as that one letter writer wrote. More people need to know about us. Brianne Barker's at Drew University, BioProf Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.